It is my privilege to introduce Sohini Ramachandran, who received her PhD at Stanford and did postdoctoral work at Harvard, and then took a faculty position at Brown University in 2010, where she subsequently ascended the ranks to full professor and is currently the Herman Focus Professor of Biology, and also the founding director of the Data Sciences. And so he has been the recipient of many accolades, and I'm not going to mention any of them, but I'll just tell you that. And she develops models and methods for understanding genetic variation. And I think there are many ways to describe the significance of Sohini's research contributions, but I'm just going to give you my personal take, which is that I see kind of two main areas that where she's generated major advances. The first one is in instructing us about how to partition genetic variation within and among human populations. Indeed, Sohini was involved in some of the first efforts to do so that were based on genomic data sets, and I think provided a model of clarity, not only in thinking about the statistical approaches that are best, but also the biological interpretations and the care that is needed when thinking about how genetic variation is partitioned, particularly among populations from different geographies. And then, about a decade ago or something, she added to her arsenal and set of subjects of interest efforts to identify the variants in the genome that affect complex traits, so-called genome-wide association studies, where I think she's been an emerging leader in helping us think about, rather than looking at it sort of one variant at a time, thinking about genetic networks and how they can help explain more of the variants of complex traits. So I'm excited to welcome.
Thank you so much. Thank you, Brett, for the invitation. I've had just a really amazingly inspiring scientific day here, and I even love the weather outside because I love winter. It's been an adaptive shift for me as a Californian moving to New England. So thanks for arranging that too. And I will say, after all the exciting evolutionary discussions I've had today, this isn't going to be the most evolutionary talk, but I think you'll see how it's motivated by being an evolutionary biologist, and hopefully in the discussion, both in questions and later with students, we could talk more about evolutionary opportunities to do evolutionary studies based on what I'm talking about today. Okay, so the question that really drew me to human genetics is this one. How has human genetic variation been shaped? How is it shaped by population histories? And I'm really fascinated by the fact that our species has a relatively deep and recent shared history, that we emerged fairly recently in evolutionary time, that our ancestors underwent large and small scale migrations, and then more recently historical mixtures during colonization and slavery that brought isolated genomes back together, and how our ancestors and their genomes dealt with changing diets and pathogens is something that really continues to fascinate me throughout my career. We've done some methodological work to contribute to the inference of deep human history, particularly some contributions to the sequentially Markovian coalescent literature with my first postdoc, Julia Palacios, who's now a faculty member at Stanford University, and we're returning to some of these questions now, particularly thinking about the X chromosome. I have a long-standing interest in the X chromosome that I always enjoy talking to Brett about, and most recently in my group, we used demographic models of sex-biased population histories, particularly during the archaic introgression events into modern humans to explain the reduction of archaic DNA that's observed on chromosome X without invoking kind of selection and hybrid incompatibility arguments. I'm also interested in how to condition on these population histories to then study the trajectory of adaptive mutations. So we might be, when we think about looking for adaptive mutations, wanting to try to isolate patterns like this, or perhaps in certain environments, certain mutations may have been beneficial. But we also know that histories will strongly influence the fate of mutations and will confound traditional tests of selection. So a big line of inquiry in my lab is thinking about how to condition on population histories to identify adaptive mutations. But if I had to summarize the concept that interests me the most as an evolutionary biologist and just kind of one idea, I would say it's linkage disequilibrium. So the way I think about genetic variation is that LD is everywhere, and that evolutionary processes leave footprints in LD across human genomes. So for the purposes of this talk, we can think about LD as just simply correlation. And I want to show some of you, because you may not have read some of these papers, what the kind of strong signatures of LD in the human genome look like. So some of my graduate work was on showing that serial bottlenecks pervade human evolutionary history, particularly in population founding events and the peopling of new continents. And these predict an increase in linkage disequilibrium with genetic divergence from Africa. So what I'm going to show you now is not my work, but work from one of the first SNP chip analyses of human population genomic data that was done by Noah Rosenberg's group in 2008 and led by Matthias Jakobsen, who's now a professor at Uppsala University in Sweden. So what you're going to see in this plot is on the y-axis a measure of genotypic LD, so basically Pearson's correlation in genotype state in 10 kilobase fixed windows across the genome, and geographic distance from East Africa being used as a proxy for genetic divergence with Africa. So this is in the Human Genome Diversity Panel, and here are the results. So what you're seeing here is that in African ancestry populations in the HGDP, you see the lowest correlation in genotype state at this fixed window size, and then this kind of systematic increase with geographic distance from Africa, with the longest shared sequences being observed in Indigenous American populations. This is a really robust signature that's been observed repeatedly, and other predictions of the serial bottleneck model since 2005. But to me, what this picture says, which I return to a lot in my research, is that LD, it lies at the heart of our identification of both adaptive and disease-causing mutations in human genetics, and we should expect that it's going to differ systematically across ancestral backgrounds. But I think we don't always, in all aspects of genetics research, account for that as we are conducting studies and interpreting the results. So as one example, I want to show you a consequence of kind of not thinking about this baseline variation in LD, which is something that I think is not often considered in genomic scans for adaptation. So it turns out if you look across different ancestral backgrounds, the number of putative signals in a selection scan will increase with LD, and the localization of the adaptive mutation becomes complicated by the footprint of LD because you get a wider footprint for positive selection. So these are results from our 2018 paper, when we apply a method called Swiffer to detect adaptive mutations just in 1,000 genomes population. So here I'm just showing two sets of results. So here you can see the scan results from individuals of European ancestry, and you see, if you can read it, which you might not be able to in the back, some kind of known suspects. So skin pigmentation genes, some immune-related genes, lactase. But then in East Asian ancestry individuals, we observe many, many more putative candidates for selection. And one could look at this and say, oh, there's just more adaptation in this set of genomes, when in fact, what's happening here is that our ability to call selection accurately is being affected by LD. And I think it's kind of interesting that actually this has been observed for over a decade in these types of studies, but I really think it's under-discussed in human population genetics. So this is the kind of complication of an LD-based pattern that I'm interested in trying to understand. But for today's talk, I'm in the middle of grant writing. And when I write grants, I'm not actually submitting to NHGRI, but as a person interested in human genetics, I like to visit these strategic priority documents they put out, which has happened every 10 years since the human genome draft sequence was published. So there was one in 2010 and there's one in 2020. And I want to talk about one part of this paper, which is a statement about bold predictions for human genomics by 2030. So there are 10 predictions that are made here, aspirations. So the idea is that we're not at this place now, and we may not get there in a decade, but these are things we should aspire to do in this decade of research. So I'm just going to quote in, I'm just quoting these a little bit randomly for my own purposes, but I urge you to look at the paper. So the first prediction is that individuals from ancestrally diverse backgrounds will benefit equitably from advances in human genomics. So again, the statement that's being made here is that this is not currently the case today. I think we can agree. Unfortunately, that is not currently the case today. But it's something that we should aspire to, and it's also something we might not achieve by 2030. Another is that research in human genomics will have moved beyond population descriptors based on historic social constructs such as race. And another is an individual's complete genome sequence along with informative annotations will, if desired, be securely and readily accessible on their smartphone. So this is an outline for the talk today. And the idea I want you to take away from this talk is that these stories I'm about to tell you are really variations on a theme of ELT. So the first 2 points are kind of the motivation for the first half of the talk, and then we'll return to this last one where we can contemplate what life would be like if we could look at our genome on our phone. All right, so obviously we have a problem in human genetics where our ability to predict complex traits and to understand the genetic basis of complex traits is such that benefits are not being realized equitably for everybody. So one problem that you're probably aware of is that in human genome-wide association studies of disease, we have lots and lots and lots of data, but we actually have less analyses of diverse genomes. So across over 6,000 GWAS studies that have been deposited in the NHGRI EBI catalog online, and there are GWASs that aren't necessarily in that catalog, only about 90% of the samples represent about 16% of the human population, and that's individuals of European ancestry. This is now an old figure from Alicia Martin in 2019, but it persists. Actually, unfortunately, periodically every decade when someone makes this point about a lack of representation in genetic studies, the subsequent GWASs get less and less diverse. So that's a thing that's happened too. But you might say to me, well, Sohini, there are all these great biobanks now. Is this really still what's happening as we look at things like the UK Biobank? And it turns out that as we push for more and more diverse data sets, often data from non-European ancestry individuals are just not analyzed when biobanks are analyzed. So this was initially pointed out in a really nice comment in Nature by Simone Gravel and co-authors, where they looked at just surveying studies of the UK Biobank and of the Health and Retirement Study, and noted how few of them provided any mention of minority populations in the study, and also that reviewers were clearly not asking for any justification for the exclusion of those minority populations. So they were just sort of not mentioned at all. In my lab, we did a scraping of PubMed abstracts that mentioned the UK Biobank and then looked through what data were analyzed in them. And this is old, but again, the end date here is 2020, but the pattern persists today that about 20% of studies at the UK Biobank will report an association outside the so-called white British cohort. But the vast majority of those are studies that are also comparing to Biobank Japan, which is a large biobank study data set where summary statistics are available from about 200,000 individuals in Japan. But again, analyzing multiple cohorts within the UK Biobank based on ancestry is actually very, very rarely done. Okay, so let's just keep that idea of lack of analysis in the background of our mind. So data is being left on the table, maybe because of differential power for analyses. And I know this is review, but just to make sure we're all on the same page, here's what a GWAS looks like. So we're going to take each SNP genome-wide and essentially just regress the state of a complex trait in our case, or some continuous phenotype onto allelic dosage. Our underlying model is a pretty simple linear model in general. Although of course there are methodological advances beyond this, and we might want to control for different covariates like ancestry-based covariates from principal components analysis or correlated phenotypes. But ultimately our inferential goal is to test whether this regression coefficient B1 is 0 or not. Another way to think about the traditional GWAS framework is as a classification problem, where essentially, as was first discussed by Bradley Efron, a statistician in about 2007, but Matthew Stevens, the statistical geneticist, has really kind of written multiple beautiful papers on this, is that the model of GWAS induces a really specific set of assumptions. So really what the model of GWAS is that there are some non-causal SNPs, and their effect sizes, which are the distributions I'm showing you here, are some point mass at 0. And any effect size drawn from a distribution with a non-zero variance is associated. The thing about this model is that if we have enough samples under this null, it looks like every SNP is associated with a trait of interest. And I would argue that in human genetics, this is kind of the world that we live in. It seems like everything is associated with height. Everything is associated with type 2 diabetes. And that's because we're asking kind of a very specific constrained question. So what this means is that our false discovery rates are really high in GWAS. So again, this concept was really beautifully encapsulated in this paper by Matthew called FDR, False Discovery Rate and Medial. But these are simulations from a former student of mine, Wei Cheng, using real genotype data from the UK Biobank and synthetic quantitative phenotypes. So under different heritabilities and different sample sizes, you can see that GWAS is a fairly well-powered approach for identifying causal mutations under this simulated ground truth. But it also identifies a lot of false positives, and that's because it's really calling almost everything as associated. So this is an issue. Right? How can we understand complex trait architecture with this method if basically as sample sizes increase, everything that's associated? So with my colleague, Lauren Crawford, who's a biostatistician at Brown and also at Microsoft Research, we wanted to alter the null hypothesis of GWAS a little bit. And we said, suppose instead of testing the idea that beta, the regression coefficient to allelic dosage in the model, is 0, we wanted to kind of test the hypothesis that beta is approximately 0. So instead of these two categories of causal and non-causal SNPs, suppose we had a third category that are interactive SNPs, which co-vary with the causal SNPs. And I guess I'm saying causal very loosely, so I mean causal or tagging and causal SNPs. But these might co-vary with these causal SNPs and have effect sizes that appear indistinguishable from some of those emitted by causal SNPs, but they don't actually directly influence the phenotype. So this alters our model a little bit to look something like this. So maybe we have this kind of salmon red distribution of weakly associated effects that aren't directly influencing our trait, and we want to differentiate the salmon from the blue. So we want to reduce that false positive rate and be like really confident about the associations that we're using for hypothesis generation. So we developed an algorithm that also relates to this paper from Nilanjan Chatterjee's group. Ours is a little more flexible, and we call it Genie. And I'll sort of gloss through a few of its features, but basically we use a shrinkage method to shrink inflated GWAS effect sizes. We do this recalibration, which is similar to kind of asking, let me just identify SNPs that are contributing to PVE in some significant way above some threshold. We decompose effect sizes into a K-component Gaussian mixture model to try to learn where that threshold is. But this is kind of the most interesting part to me, the LD part, which is that we then take the SNP level effect sizes and use LD in the region in order to compute, using the covariance structure among SNPs, what we might expect a gene level association score to look like in that region. So I'll show you what these kind of conceptual advances do for false discovery rate here. So this is the original GWAS plot that I was showing you, and I'm going to show you something where our method does really well, but I'm going to show you other methods that also do well. So some of these you might have heard of, and the SCAT family of tests that was developed with the Broad Institute, and then RSS, which is from Matthew Stevens. So what's happening conceptually here to reduce false discovery rate is that What the SCAT family of tests is doing is it's kind of doing a burden test. So it's aggregating over these SNP level GWAS results. RSS is then shrinking the inflated GWAS effect sizes, which really reduces your false discovery rate. But that last little bit of false discovery can get controlled really well by then modeling LD on top of it. So we did this methodological work, but the motivation for it was getting back to this question of how can we try to analyze biobanks that have differential sample sizes when we do GWAS? And could going for gene level association tests help us deal with the reduced power in these imbalanced sample sizes? But I think the biological question that was interesting to us to ask at the time we were doing this study is, given that LD differs across ancestries or maybe other partitions of individuals, but for now we'll talk about ancestry, how should we conduct multi-cohort GWAS studies? And I would say right now, we're asking a very specific question in GWAS. And the model that underlies it is we're trying to identify mutations where the true mutation level effect on trait architecture is going to be the same for all individuals. Or maybe not the effect, but that the architecture itself at the mutation level is the same. But you can imagine that this is just one model for trait architecture, right? Traits can be more complex than this. So we could also imagine maybe that there are some traits where they're underwritten by a set of core genes, and those could be the same across everyone, but with varying causal mutations. Maybe different perturbations to the system can produce the same trait. You could imagine that the core genes differ across ancestries, but they're all in the same annotative pathway, or just for completeness, something totally different. So the complement of all these other things. And the truth is that often in classes we teach, or if you read around in the literature, there are lots of examples of each of these models of trait architecture, right? So one size doesn't fit all of trait architecture, and we can think about and see some evidence for each of these. So we were interested in trying to find ways to test a kind of nested set of hypotheses, and this resulted in this 2022 paper in ADHD. It was a really interesting paper for my group at the time because actually every member of the group is a co-author on the paper, which was really fun. It was led by a former grad student of mine named Sam Smith. And Sam really did a kind of heroic job bringing together multiple biobanks. So the UK Biobank, Biobank Japan, and then we had a manuscript agreement with the PAGE Consortium, which is a large study of admixed individuals in the US. We also collaborated with a former colleague of mine, Ben Raphael, who's now at Princeton. And the idea here was to take the gene level association scores and then feed them into gene networks in a kind of graphical model to identify connected subcomponents of these graphs that are enriched for strong gene level association. So basically asking, are the genes that seem to be strongly associated with these traits also interacting with each other in ways we wouldn't expect based on chance? So I just want to show you one kind of result that illustrates what I think this kind of approach can let us see. So we found when we were looking at associations with triglyceride levels, this one large gene subnetwork of apolipoproteins that was enriched for association with triglycerides across multiple cohorts. And the kind of traditional way to look at this, along with our sort of protein-protein interaction network results from Ben's group, was to sort of look at, okay, here's the white British cohort, it's got a lot of power, identify these genes that are sort of known targets that might be influencing triglycerides. In Biobank Japan, I see some overlap in these signals, but it's sort of slightly different part of the graph. In fact, in Biobank Japan, we identify a sort of ancestry-specific association with this gene BASE1, which has been identified as associated with triglycerides and Alzheimer's disease in multiple studies of East Asians. BASE1 encodes a peptidase A1 family of aspartic proteases that seems to influence this phenotype and is highly expressed in the number of relevant cells. And then in a really small cohort in the PAGE study, we also see multiple gene level associations and another kind of ancestry specific set of associations with some alpha globin genes that have been seen to be associated with diabetes in Hawaiian ancestry individuals who have triglycerides usually as a comorbidity. So this is one way to think about doing an association study. But another way is to kind of pull all these together jointly in a single analysis. And then what we see is kind of a more holistic picture where in fact, all of these genes are connected to each other based on this PPI, which we can argue about how much we believe PPI, but in fact, it seems like these native Hawaiian genes are actually in really long regions of LD and appear kind of ancillary to this network. But I think that if we had larger sample sizes, we might find associations within the kind of core of this network. But I guess to me, what this says is it's kind of an anecdote about how thinking about multi-ancestry data, I think the way we're doing it now, it's a little bit like the adage of the blind man and the elephant, where everyone's kind of feeling around a different part of the same thing without sort of looking jointly. And so I think that finding ways to not leave data on the table, particularly data that brings us more LD diversity into analysis and that models genetic heterogeneity and causal mutations, can let us identify both methods sensitive to ancestry-specific alterations, but more importantly, actually highlights the shared nature of genetic trait architecture across people. We've also tried to look at whether regions that are rich for contributions to heritability overlap across ancestry. So here's just an example of this, looking at urate levels. So urate, the synthesis and transport of urate is a well-known thing in biology and biochemistry. So here what we're doing is a heritability analysis using Hess from Bogdan Kosinuk's group, and we're just asking if some of the significantly enriched regions with heritability, significant contributing regions are enriched for being these known transporters and synthesizers of urate. So this definitely seems to be the case in individuals of European ancestries, which Jonathan Pickford's group had already demonstrated. And when we look across different ancestries, you see a lot of overlap in the signals, as well as here shown in blue and here shown in red, some putative ancestry-specific enrichments. And again, even in pretty small cohorts, we can at least see the strongest signal in the largest cohort replicated in this known solute transporter. In my group, where we're going with this now is to do multivariate GWAS studies. And so just to show you what some of this might look like for the future, these are Gini results looking at LDL and HDL levels in the UK Biobank. Some of the genes are sort of annotated. There isn't a lot of overlap in signals, which maybe we would expect because these are slightly different traits. But when we look at these and try to identify those multivariate Gaussian components in this bivariate space, what you see here is a lot of univariate associations with this that are different, right? So maybe if we want to control for pleiotropy, say, as we try to prioritize genes, this kind of analysis could be helpful. But we also see some shared targets of both weak and moderate effects. And so digging into these is the next thing we're doing in this space. So some of the questions that I think come out of this for us are, how can we try to understand pleiotropy better versus trait-specific associations by using gene-level types of tests? And the funny part about this work for me is by the end of Sam's paper, when it came out a couple of years ago, I sort of realized that actually ancestry is like the least interesting way to partition people. And I think that what is going to be more interesting for us is to think about alternative partitionings of the data that can highlight gene by environment interactions underlying traits. So for example, if we looked at the heritability of heart disease in different age cohorts in the UK Biobank, we would see different heritabilities because of differential interventions. And we could look at things like gene by sex interactions in this multivariate set of methods, which I think will be really useful. But the point I also want to leave you with is that if we really want to have all people benefit equitably from research in medical genomics, I think that we need more than just data. We also need methods that can analyze data, especially data that's really, really imbalanced. So that's one of the questions that I'm interested in. Okay, so now I want to switch gears completely, and I want to tell you about some work that's unpublished. And I'll say that talking about unpublished work in a talk, it's a little bit like how a comedian must feel when they're sort of trying out new material. So, I welcome feedback, no booing, please. But okay, so I think this idea is kind of interesting. Like, what world would I live in where I want to look at my genome on my phone? But it does make me think about biobanks. And I think an interesting feature of biobanks is that They are full of relatives. So biobanks contain lots of genetic relatives. Many of them are distant, although maybe not as distant as you would think, like on the level of fourth cousins is what I'm largely going to talk about today. And increasingly close relatives are increasing in number as sample sizes increase, particularly in biobanks that have a goal of sampling a certain proportion of a population. So I'm going to show you theory from Rasmus Nielsen's group about this, which turns out to bear out in a couple of biobanks. So here what's being shown is the proportion of a population that's sampled in a biobank on the x-axis and predictions of the proportion of biobank participants that will have a relative of a certain degree. So first degree being a parent, child, or full sibling. You can think of fourth degree as a first cousin, sixth degree as a second cousin. So what this is saying is if a goal of a biobank is to sample, say, 10% of the population, then about 10% of the biobank is going to have a first degree relative in that data set, and everybody's going to have 8 cousins in that data set. And just as an example, FinnGen, the Finnish biobank, has sequenced genomes of 10% of the Finnish population, and of these, 30% are first degree relatives. Okay, so this is a kind of lower bound on that estimate. But it can obviously be elevated due to population histories. Okay, so what that means is biobanks are giving us new opportunities to study identity by descent and to think about population genetic inference from IBD. So what is IBD? An IBD segment we can think of as an approximately identical region of the genome that 2 individuals have inherited from a recent common ancestor. So in this image here, this part of this dark green segment was inherited IBD from this maternal grandparent of these 2 individuals here at the bottom. And IBD segments are initially very large, but they successively decay in size with each generation, with each meiosis. So if we kind of focus in on this IBD segment a little bit more, we can also think about how IBD sharing might be distributed across the genome. So the human genome is approximately 3,500 centimorgans long. And what I'm showing you here is a kind of putative distribution between a focal individual and their grandparent of IBD sharing. So these individuals are going to share 12.5% of their genome IBD, but some of these segments will be short and some of them will be very long, right? So even maybe as long as 95 centimorgans. The total sum of IBD sharing here is going to be, in this case, 1,973 centimorgans. Sorry, I said 12.5%. That's totally wrong. Anyway, that is the answer. Distant relatives will be much less likely to share IBD segments. So this is the proportion of pairs of particular relationships from first cousins to 8th cousins who share at least one segment IBD that's more than 5 centimorgans long. So first cousins will definitely share that, right? And first cousins once removed and so on, but it will decay and decay and be pretty unlikely for 8th cousins. But they might share one segment of that. Still, when you have a scale of data, you may have a lot of these 8th cousins. And that's in fact the case. So if you think about IBD sharing in the UK Biobank, which has about 500,000 residents in the UK, and we're talking about this white British cohort again. So they're meant to, or they represent approximately 1% of the total white British population of the UK. The average participant in this cohort shares at least 5 centimorgans IBD with 24,000 other individuals with a fairly large standard deviation. And they share at least 35 centimorgans total, the equivalent of second cousins with about 23 other individuals, with again, a pretty large standard deviation. So in this biobank, there are some large families that are present of pretty recent relatedness. Okay. So there are lots of things that I think are really interesting to do with this data. The first thing, which I'm not really going to talk about today, is that often in population genetics, we just throw out like 3rd degree relatives, 2nd degree relatives from inference. But when you're talking about throwing out, you know, a large proportion of your data set, maybe there are other ways to get around that. And I can talk about that later in questions. But we had a simple goal in thinking about this question of IBD sharing, which is to say, okay, if we have IBD sharing, what if we could classify IBD segments an individual shares with their relatives by the parent of origin through which they inherited those IBD segments. So we call this trying to pull apart the two sides of the family. So I'm trying to paint chromosomes based on IBD and then ask, I'm related to these individuals, but how am I related to them? Is it through my mother or through my father? So why might we be interested in this? What are the downstream applications of understanding the parent of origin for IBD segments? Well, the one I'm going to tell you about today is that it enables interchromosomal phasing. So to date, really, statistical phasing just talks about phasing individual chromosomes. So this is trying to recover which allele was inherited on which chromosome arm, right? So it doesn't stitch across the chromosomes. So we can't really connect haplotype 1 from chromosome 1 to maternal or paternal haplotype on chromosome 2. But the other things we could do then is we could do parent of origin effect GWAS studies. We could also start to ask questions about polygenic indices, the maternal versus the paternal contribution to them. And I think a really interesting population genetic question we could answer is parent-specific local ancestry influence. So this is from a 23andMe blog. So this is a current or former 23andMe employee. And some of you probably know that 23andMe gives this kind of output based on local ancestry and the length of IBD segments or tract lengths, the timing of that ancestry. But now it's given to you without a sense of which part of the family it comes from. You could imagine that with parent of origin assignment of these tracts, you could actually start to answer that question, like what's maternal, what's paternal. Okay, so this is work by my current student, Cole Williams, who is really interested in all things identity by descent. And so he's come up with an algorithm for bipartite clustering of IBD segments. So how do we pull IBD segments into these two clusters that represent the two sides of the family? The idea here is to represent IBD segments as nodes in a graph that have been inferred after regular phasing. So let's assume we have the scale of data, which I believe we have, and we're going to demonstrate that in our paper, that we believe are intrachromosomal phase. Now we're going to take that and represent IBD segments in a graph where positively weighted edges between segments indicate inheritance from the same parent, and negatively weighted edges between IBD segments indicate inheritance from different parents because they're on different haplotypes. And so we can do bipartite clustering on this graph using spectral clustering on the signed Laplacian of the graph. And this has been a really interesting project because this is actually in computer science what's called an NP-hard problem on a signed graph to do this kind of clustering. But we found a paper that did it, and it offers a really interesting heuristic for if the graph even shows two clusters based on the leading eigenvalue from this signed Laplacian. So I'll come back to that in a little bit, why that eigenvalue matters. But the paper that published this had a really interesting use case, which you might like as biologists. So what they did is they had this data on relationships between groups in Papua New Guinea. And so the green edges here represent friendly relationships or interactions. They're the positive weights. And the red edges represented active fighting between groups. So the reason why using the negative edges can be useful is it lets you implement the idea that, not to be too cute about it, but like that the enemy of your enemy might be your friend, right? So our goal is to do this kind of clustering on IBDs. So I'll just show you a little bit of what that looks like. Okay, so let's imagine we begin with statistical phasing that we've done with some state-of-the-art method. And even suppose that we have a switch error in that phasing. So we can be robust to that actually with this clustering. It can help us identify switch errors. So we've done the phasing. And now what we're doing for this focal individual is we're calling all IBD segments that are more than 5 centimorgans for all other individuals in the biobank. So the letters here represent different relatives that the individual is sharing these segments with IBD. Okay. So then what we're going to do is collapse these co-located IBD segments into what we call tiles. And then we have subtiles depending on the original intrachromosomal haplotype assignment. So same tile IBD segments on different haplotypes are in different subtiles. And we're going to begin by assuming the IBD segments that are on different haplotypes were inherited from different parents. Okay. So now what we can do is also add the fact that for some relatives, we observe more than one segment inherited IBD. Like H here, there's one IBD segment here on this subtile and another here on this subtile. Similarly, we have J2 and J1. Okay, so the mixture of some close relatives, but some distant relatives that produces lots of IBD segments throughout the genome is what's going to let you do that interchromosomal phasing. So then we can create this IBD graph. We have these positively weighted edges between nodes on the same subtile, and then the negatively weighted edges to force apart the separate haplotypes. And we create an IBD graph. We add weights for the same relative as well here, so connecting J1 and J2, H1 and H2. And then we do the bipartite testing. So now we're saying, okay, putatively, all of these segments are coming from one side of the family and all of these segments are coming from the other side of the family. And that leading eigenvalue of the sign Laplacian, if there are lots of these cross connections, like H1 to H2, then that eigenvalue is going to be really small, which means the clustering isn't working. And I'll tell you why that might happen in a second. But this has been working well for us in the UK Biobank. The way we're able to test it is using trios. So there are 1,000 white British trios in the data set. I'm not sure. I was surprised to see how many trios there were because they weren't actually sampling for them. There are a ton of full fibs as well. So what we do there is we're trying to do this clustering by excluding the parents. So now we're just using the distant relatives, and then we're using the parental phase to check our clustering answers. So this precision plot's a little bit hard to understand, but I'll spend some time on it. What this is showing us is that as we try to recover IBD with each parent correctly, leaving parents out, we want to see how much of the genome that we're covering. And it turns out that if you look at the precision here, we're able to cover a lot of the genome for, almost a full genome for many individuals, but at pretty low, well, I don't know. Actually, I think this is high precision, but the median value here is 70%. For some individuals, though, they have very few relatives who are not their parents in the data set. And that's why we recover only a little bit of their genome, but we can do that with very, very high precision. So this is also going to vary across ancestries depending on the representation of the broader families. But there are quite a few individuals up in here where we can essentially identify, like repaint their whole genome with IBD without including their parents. So here what we're doing is we're adding all possible edges, even with like those 5 centimorgan IBD segments. But you might say, okay, let's use a kind of heuristic based on total IBD sharing, where we say maybe we want like maybe at least 10 centimorgans of IBD sharing total before we include individuals in the graph. So then that improves our precision highly, but we cluster less of the genome. So on average now we're clustering maybe half the genome correctly, but again, we're doing it like highly accurate. Okay, so why should we care? Well, I think there are sort of two really fascinating fine scale population structure questions here. And they go back to an old question from my postdoc research on the actual pedigree of family relationships and how those constrain coalescent events. So this is another kind of 23andMe graph. This is not my ancestry timeline. This is a putative one. But basically, I think if we knew the parent of origin of these tracks, it would actually change our inference of the gene flow history. If, for example, I tell you, you might think this is maternal ancestry, let's say it seems parsimonious at all, this is European and this is like paternal ancestry, or one parent or the other. But what if I told you this person had two parents who consider themselves half Japanese? Now, kind of the sense of the migration history in that family pedigree is totally different. So that's what we're trying to explore with this. But the other thing that bipartite clustering gets us is these, we're calling them purple nodes. So I'll call H here a purple node. So it's an individual who shares IBD with the focal individual through both sides of the family. And there are these individuals. So if you have, say, a third cousin, fourth cousin, genetically, that person may be related to you through both sides of the family. And so actually, we're finding that we can use this to identify rates of pedigree collapse and how those might vary in different diaspora waves in different biobank data sets. So in this vignette, I think that there's a lot that we could do if we could understand a parent of origin for IBD segments in biobanks, and particularly for local ancestry tracts. I think this will also, as we think about IBD, let us condition on recent relatedness in biobanks to see how it affects our inference of effective population size in the past tens of generations. And that there are some, I think, really interesting inter-chromosomal phasing applications and opportunities that biobanks are offering us because of their scale of data. So these are the group of people that I'm really privileged to work with today. I told you a little bit about Wei and Sam and Cole's work and Sharon's as well. And I would be really happy to take any questions that you have. Yes. Yeah, so the question is, have we done any parent of origin GWAS? So we have not, but the developers of Shape It have. So Robin Hofmeister is the first author of a paper in, I think, 2022 in UK Biobank, and they basically replicate, I think, parent of origin effects in 6 different quantitative traits in UK Biobank, as well as 1 novel association. So they demonstrate that it can be done. Our plan is to do it in the All of Us research program. There are also some interesting questions to ask, like with our approach, across chromosomes, are maternal versus paternal effects, how much do they differ in like their sign and contribution to a trait? So do the maternal effects for certain traits tend to be trait increasing versus trait decreasing or something like that? So I think there's a lot of interesting, there are a lot of interesting questions to ask there, but yeah, they demonstrate that it can be done. Or just imprinting effects, right? So, there may be like growth promoting, parental effects that are paternally expressed, but reducing ones that are maternally expressed, something like that. So there's like a whole literature on sort of imprinting that talks about some of these loci, but probably others in the audience know about better examples than I do. Yeah.
Performing the direct analysis where the null hypothesis is not fitting to be located approximately zero. So are the methods that we use for studying the epsilon have not been very further specific to the program?
Yeah, so that's a good question. So then talking about Jeannie, kind of how do we get to this approximately zero approach for the null hypothesis? I'm repeating for the Zoom. Was that a fair assessment of the question? So we did kind of a clunky way and then a sort of grid search approach under simulation. So essentially we found that for our purposes, we were trying to control we really wanted to isolate the strongest associations from weaker associations. So we started by saying, what if we just kind of looked at the variance of the second component or the third component as being that epsilon threshold? But then now we have implemented as part of the package a way to like learn that epsilon genetic threshold based on kind of a regularized like local approach.
I've looked at the paper because it seems like a very flat application.
Yeah, our hope is that it limits spurious confounding. But it does mean that you're maybe also increasing your false negative rate depending on how strongly you set that epsilon. Yes. Mhm. Yeah. Yeah, so in that result on the apolipoprotein network associated with triglycerides and the native Hawaiian cohort, right, so we identified ancestry-specific associations, but then I made a comment about LD as being part of why we might have identified that. And so what I was trying to say, it's a good question. So definitely, you know, in the paper we talk about, like, with some traits, we see evidence of pretty heterogeneous effect sizes across ancestries.